Welcome to the latest installment of the AJT Journal Club series. Today's Journal Club on the AJT article, First Clinical Grade Porcine Kidney Xenotransplant Using a Human Decedent Model, is hosted jointly by the AST Education Committee and the AST Kidney Pancreas Community of Practice. I will now turn the session over to our moderators, Dr. Andrew Adams from the University of Minnesota and Dr. Burcin Ekser from Indiana University to begin our presentation.
So thank you, Brian. On behalf of my co-moderator, Dr. Burcin Ekser from Indiana University, wanted to welcome everyone and for joining to this AST Journal Club, where we'll be highlighting some of the most recent exciting work on the field of xenotransplantation. As many of you are aware, there have been recent reports of significant advances in preclinical models of xenotransplant. Even more importantly, in the last 6 months, there's been several landmark studies focusing on its clinical translation. And today, we're happy to have two leading surgeon scientists in our field from the University of Alabama at Birmingham. And they're going to discuss the first report of a clinical grade porcine kidney in a human decedent model, as well as additional work that's been done by Dr. Montgomery and his team at NYU. Just highlighting the advances in the field that are occurring even month by month. So it's my pleasure to introduce Dr. Jayme Locke first, who is a professor of surgery and director of the Comprehensive Transplant Center and chief of the Division of Transplant Surgery at the University of Alabama, Birmingham. She is an NIH-funded surgeon scientist who is internationally recognized for her work in transplant outcomes and health disparities. She's the deputy editor for the American Journal of Transplantation and a counselor at large for the American Society of Transplant Surgeons. She's received numerous awards and accolades, but I think most of all, just knowing Jayme, she's a true advocate for her patients, including those who are disadvantaged and have reduced access to care. And I think that's driven in large part her interest for xenotransplant and providing a therapy for those that might not otherwise have access to kidney transplant. I'll turn some time over now to Burcin to introduce Dr. Porrett.
Thank you, Andrew. I also like to welcome and thank American Society of Transplantation and AJT for this journal club. We are very excited because we are going to discuss this first clinical grade porcine kidney xenotransplant model. And definitely the AST Education Committee and Kidney Pancreas Community of Practice put a lot of effort. And our second panelist is Dr. Porrett. So I am very honored to present her. That Dr. Porrett is Director of VCA Transplantation and Director of Clinical and Translational Research at UAB. She is nationally and internationally known for her studies on uterus transplantation previously. Definitely she's famous now on xenotransplantation field as well with this recent publication. Her laboratory is NIH funded in immunology, especially transplant immunology, and we are excited now to hear what they will tell us. So thank you, Dr. Porrett and Dr. Locke.
Great, well, thank you both so much. We're really excited to be here, and thank you all for joining us. So I'm Jayme Locke, and joining me is my colleague, Paige Porrett. We're going to tag team this presentation today and try to put our work in the context not only of what we did, but the recent paper that many of you may have seen that came out from the New England Journal late last evening. So here are Dr. Adams' conflicts, and then Dr. Porrett and I, we received funding from United Therapeutics and its subsidiaries, which include Lung Biotechnology and Revivicor, and we will be discussing the investigational use of both the U-kidney and the U-thymo kidney. So with that, I thought I would jump right into sort of, why does this matter particularly to me? I am, of course, a health services researcher that's really focused my career on health disparities. And that's really, as Andrew put it, is really what's drawn me to the field of xenotransplantation. I think as everyone on this call is very familiar with, the end-stage kidney disease burden in the United States is substantial. There are roughly about 800,000 Americans currently living with end-stage kidney disease. And we know that racial disparities exist within that disease burden, such that African-American or Black individuals, independent of their age or sex, have a higher incidence rate of end-stage kidney disease compared to other races, including whites, Native Americans, and Asians. And so ethnic minorities are, in fact, disproportionately impacted by this disease. And importantly, the greatest disease burden actually exists in the southeastern United States. And you can see that here with the darker orange kind of occurring through here. And it kind of really overlaps what you may have heard of being called the stroke belt. This is what we would call the end-stage kidney disease belt, and was first described by one of our colleagues here at UAB, Dr. Rhiannon Reed. And what's important to note about that is that if you look at those states in the southeastern United States and compare it, for example, to a state in the northeastern part of the US, you can see that the percent state population that is African American is quite large compared to the overall US population and to, for example, the state of New York. And importantly, our disease burden is also higher. And what's important about this is that we know that end-stage disease burden not only drives demand, but it also drives supply. So in areas that have high need for a kidney, they also have lower access to organs. And this is some work done by one of our partners, Dr. Cannon, in which he looked at transplant rates versus end-stage kidney disease burden using some geospatial analytics. And what you can see in the sort of royal blue colors, these are areas that have low transplant rates, but high end-stage kidney disease rates. And so you can see that's right where we live. Here's the state of Alabama. And these little blue areas really overlap with that end-stage kidney disease belt that I described. And so without question, we have a huge gap between those in need and the supply of organs available. And we all know this. Many of us have seen this curve a million times. Our transplant curve has really been flat for the last 20 years, despite our efforts to increase. We know that 40% of waitlisted patients, which are really sort of the healthiest of our end-stage kidney disease patients, even among those, 40% will be dead within 5 years. And so really the organ shortage is a lethal problem. It's refractory, it's severe, and it really needs a radical solution. And I think this is really where the field of xenotransplantation plays a key role. And it's really the impossible solution, making it the possible solution to the organ shortage. I do want to just acknowledge Martine Rothblatt, who's the chairwoman for United Therapeutics, and her role in this and the longstanding partnership we've had with her here at UAB. That dates back to 2016 when we first partnered together to build and develop a pathogen-free facility and really work towards taking all the work that folks like Andrew have done in the laboratory and really bring it to the clinic for our patients. And hopefully xenotransplantation can be a solution to the health disparities that I just described. And that certainly is my motivation for wanting to be involved. So what we know about xenotransplantation and the genetic modifications that have occurred to date in the porcine source animal is that really our goal has been to improve compatibility in humans, not necessarily non-human primates. And I do want to acknowledge the work of many people, including our moderators, who have really dedicated their careers to xenotransplantation and have studied this for many years in the context of non-human primates. And it really, without their efforts and work, I don't think the developments that have happened in the last 6 months would ever have come to light. So just really want to acknowledge that. But let me walk you through this slide for a moment. So up here is looking at human sera, and you're looking at IgG on the x-axis and IgM on the y. And the goal is to get these little cells down here into that left lower box. And that would suggest that you would have a negative cross match. So this is what human sera looks like against a wild type pig. And then as it marches through the various knockouts of the xenoantigens on the pig graft, and you can see that it really takes 3 before you start to see some compatibility with human sera. And so it's really the triple knockout of the xenoantigens that is the best and demonstrates the best compatibility with humans. You can see that shown here. But what's interesting is you never get great compatibility with the non-human primate model, whether you're talking about baboons, rhesus monkeys, or other types of monkeys. And so it really suggests that the non-human primate model, for all intents and purposes, is really a model of incompatible transplantation. And that tells you how difficult this has been to study in the lab. And so how do we get the remaining pieces of information when we have sort of an imperfect model? Is there another model that we could potentially use? And there are critical knowledge gaps. So certainly the pig has been genetically engineered to look less foreign to the human immune system. It's been extensively tested in non-human primate models and been shown to be able to help a non-human primate survive after the native kidneys have been removed. So it's been able to demonstrate that it can maintain electrolyte homeostasis, it can produce urine, it can help the animal survive. But there are critical gaps that remain, and that's because, as I just mentioned, the non-human primate is in fact immunologically different from a human, and we need to be able to test this model using conventional immunosuppression that we already use in human-to-human allotransplantation. So how might we do that? And I think it really involves trying to develop a preclinical human model or a model of brain death that we have called the Parson's model. And this was after our first donor family that participated in our study. And I think it's important to note, this is actually an idea that Tom Starzl had some 30 years ago. And it really made sense to us that could we use a preclinical human model of brain death or the Parson's model to answer some of these remaining questions that we felt really needed to be answered before we would feel comfortable moving this into a living person. So the design advantages of using brain death as a preclinical human model is that we could answer these questions without actually harming a living person. We could test our engineering in a human before the living, and we could test our immunosuppression. And I highlight this slide because this really has been an incredibly collaborative effort that involves not only UAB and United Therapeutics, but also our partners at our organ procurement organization, Legacy of Hope, as well as UAB Medicine. And that is really how the Parson's model came to be. And so with that, we were really excited to see the publication from last evening, to see that the Parson's model has been leveraged by an additional group that's been able to do 2 additional cases of pig-to-human kidney xenotransplantation. And we were approached by the board to really offer how our study compares to this recent publication. And so with that, I thought I'd turn it over to Dr. Porrett, who will really highlight what our study accomplished, as well as what Dr. Montgomery's study accomplished, and what the remaining knowledge gaps are. So Paige.
I want to thank our moderators, the AST, and of course our attendees again today for the privilege of presenting both our work as well as to compare and contrast to this really critical publication that was just released yesterday in the New England Journal of Medicine from the NYU group. I know this publication as well has been long awaited, so I appreciate your indulgence as we walk you through, I think, a couple of the key knowledge gaps that both of these studies have addressed. And so the first thing that I wanted to highlight was that, very importantly, both of these studies have demonstrated in a human being that there is an absent hyperacute rejection event. And I probably do not need to introduce the audience to the fact that, again, based on Dr. Adams and others' works, we have known that there is a tremendous barrier to the transplantation of a porcine kidney into non-human primates as well as humans. And so I'm delighted to tell you that both studies, as I've represented here with these pictures, photographs, these figures from the 2 publications, that all 3 transplant recipients did not have hyperacute rejection. And you can probably, it's probably difficult to see this here, but I just want to emphasize that Dr. Montgomery and Dr. Stewart's study showcases the results of 2 recipients. The top recipient is shown here, both the kidney appearance right after reperfusion, as well as the recipient at 54 hours. The bottom row shows the recipient results after reperfusion again, and this is the second recipient again at 54 hours. So a clear long time frame of many, many hours has passed, and there's no hyperacute rejection. Similarly, in our study published some time ago in One Decedent, I'm showing you the results of both the right and the left kidney. And I should mention that we transplanted both kidneys into our Decedent, and I believe that Dr. Montgomery and both transplant recipients transplanted an individual kidney. So this is, again, one Decedent, 2 kidneys, that you can see the same result, which is no hyperacute rejection, followed many hours later, in our case at 72 hours, with persistent viability of the transplant. So this is a very key safety finding that will help propel the field forward. The next knowledge gap that I want to highlight is, as you can read on this slide, is that we think that our study has actually now really answered this knowledge gap and demonstrated that the pre-transplant cross-match predicts compatibility. It's probably not a surprise to this transplant audience that that would be the case, because we know of the critical importance and requirement for performance of a prospective cross-match to predict compatibility in allotransplantation. And our center took a very similar approach. I invested quite a bit of time and resources in the development of a prospective cross-match that was performed in this case before the transplant was administered. Very briefly here, I'm hoping this coming through your Zooms for everyone. You can see the results of negative control, positive control, and then our Decedent studies. Again, this was performed prospectively as well as retrospectively after transplant. And you can see, based on our positive and negative controls, that our Decedent recipient had a negative prospective cross-match. Obviously, this informs the ability to do transplantation. At least we think that it does. And Dr. Montgomery's group I want to highlight here also did some cross-match immunologic testing that is very important. But I think that the knowledge gap that now persists or remains really stems around the predictive ability of the real-time endothelial cross-match, which is highlighted in their results here, to actually have predict the presence of either compatible or incompatible transplant. And so in short, what is being shown here on the left are the results of their IgM and their IgG flow cross-match binding assay. You can see in the black, there is a positive control that's included. In the green and the red are their different recipients. And I want to highlight here that unfortunately, there's not the inclusion of a negative control. There is also, I know that you cannot read the y-axis on these slides, but this is the median channel shift in fluorescence. And this result here reads 5,000. We're not quite sure, based on this publication, what the threshold would be to call a positive versus a negative cross-match. But this is still a pretty significant shift in both their recipients in terms of the flow cross-match binding, both for IgM and IgG down below. This group performed a confirmatory cytotoxicity assay that is what is being referred to here as the real-time endothelial cross-match. And you can see that both in the recipient and more significantly in the second recipient of the porcine xenografts, that they do have some positive cytotoxicity. And that's really contrast with the complement-only negative control that's shown here, as well as with the positive control that they supplied. So our interpretation of these results are that this is an incompatible transplant, and it really remains to be seen why no hyperacute rejection was discovered here in light of the cross-match and antibody binding assay that's shown. One last comment I would make is that it's unclear from the publication whether these results were performed or acquired prospectively before the transplant was performed. But in personal communication that Dr. Locke and I received from Dr. Montgomery's group some time ago, our understanding, at least for the first decedent in their studies, was that this was performed retrospectively. Another major question that everyone in this audience wants answered, as do we, is whether the porcine kidney xenotransplant is going to perform, and whether it's going to make urine, whether it's going to clear creatinine and ultimately be a rescue from dialysis. And unfortunately, this is a knowledge gap despite these two studies that still persists. Because as you can see over here in our study on the left, as well as Dr. Montgomery's study on the right, that really neither xenotransplant or set of xenotransplants effectively cleared creatinine, at least not in a definitive, clear-cut way. And so just to highlight this, in our Parsons model, the native kidneys are removed. And you can see, again, I apologize because I'm sure the axis is a little blurry for you, but from both the right kidney shown here in green and the left kidney here shown in purple, this is the cumulative urine output. So we were able to have urine output from both kidneys in our study. And again, as published, we noted that the serum creatinine in our Parsons model unfortunately never cleared. It continued to mount and rise over the course of roughly the 72 hours of the duration of the experiment. Again, we emphasize that we removed both kidneys, which is being demonstrated here as was in the publication. with pictures of the renal phosphate bilaterally to illustrate that we had taken them out prior to transplantation. In Dr. Montgomery's model, the native kidneys are present, and although from the time of transplant, which is shown at this dotted line in both recipient 1 and recipient 2, you can see that the kinetic eGFR, which is portrayed here with these this line and the triangles that I'm indicating, that GFR increased after the time of transplant. But as acknowledged by Dr. Montgomery in the publication, as you can see from this direct quote here, that it was difficult to interpret this experiment, and the conclusions about isolated xenograft function cannot be made from these data, specifically because the native kidneys were still present. So this is certainly suggestive that there was function of the xenotransplant, which is not something, or in terms of function, specifically creatinine clearance that we saw in our model. But again, it's difficult to know exactly how much the xenotransplant in this setting contributed to specifically creatinine clearance. But once again, both in Dr. Montgomery's study as well as ours, we absolutely see urine output. And I think this just emphasizes what many of our nephrology colleagues already know, which is that urine output does not equal creatinine clearance. I'm going to go through some of the similarities and differences in the biopsy results from both these studies. What we saw, and I will not walk through all of these panels today in the interest of time, but of course, please do see the paper for more detail. But we found evidence of thrombotic microangiopathy in our Decedent kidneys bilaterally. Not the Decedent, in the Porcine xenotransplant bilaterally. Importantly, we found the TMA on H&E stain. What we did not see was any C4D deposition, and you can see here again in the top row, we have a day 0 control pre-implantation biopsy, so that when we interpret the nonspecific staining in C4D by immunohistochemistry down here at day 3, and at the time of explant, there's again nonspecific staining. We did not see any C4D changes. What is not shown here in the interest of time is that we performed immunofluorescence microscopy to assay for both other types of complement deposition as well as IgG and IgM, and we did not see any of those particular antibodies or complement binding in our histologic analyses. So we are still at a loss to explain why we are seeing the TMA in our Decedent, but can speculate that it is either due to the alternative complement pathway, which may be activated in the absence of antibody binding in the setting of this compatible transplant, or perhaps due to the TMA may be due to non-complement mediated histologic damage in the kidney, yet to be determined. In Dr. Montgomery's study, again, they had improved findings on H&E. I'm afraid that I don't know exactly when these biopsies were taken during the course of their transplant, but both in recipient 1 and recipient 2, they did not report any TMA whatsoever, and the histologic appearance of the kidneys was quite good. Below in panel C and D here, you can see the results of their C4D staining, and in contrast to our study, they used immunofluorescence microscopy, so that explains why you're seeing C4D staining in 2 studies that looks a little bit different. But whereas in recipient 1, again, although there's no negative or positive controls shown here, it looks like the C4D staining is absent in recipient 1, but is present in recipient 2. And I'll mention again, data I'm not showing you here, but was included in the publication, is that in recipient 2, they have reported, they followed up on the C4D positive findings with electron micrographs that reportedly do not show any type of podocyte effacement and normal glomerular architecture. So we have differences in between the 2 studies, and ultimately are not currently able to explain the etiology of the histologic changes, either in our study or that in Dr. Montgomery's study. However, so this is, I call it a persistent knowledge gap. I want to move on to, again, a comparison and contrasting the 2 studies with respect to the type of immunomodulation strategies that were employed. In our study, as you can see here in table 5, we employed what Dr. Kirk in an editorial at the AJT has referred to as vanilla conventional immunosuppression. And in short, we used induction therapy with thymoglobulin as well as rituximab, We also included a CNI being tacrolimus, as well as mycophenolate mofetil and methylprednisolone for our maintenance therapy for the duration of the experiment. Meanwhile, in Dr. Montgomery's study, they chose a different approach, which was to use the euthymal kidney. And in this model, what they have done is to actually to surgically implant in the xenograft porcine thymus, which is what is shown here in the micrographs. And they chose this option because, as is shown in the excerpt from their paper here, that thymokidneys can promote tolerance. At least that has been suggested in animal models that they cite in the paper. Of note, they did use some immunosuppression agents, specifically MMF and steroids, but we can conclude that given the absence of tacrolimus in particular, or call it a more targeted T-cell agent, that they were hopeful that in the absence of such agents that some type of immunomodulation would be conferred on the T-cell compartment from the thymokidneys. Finally, I wanted to talk about, well, not finally, almost finally, the impact of the Parsons model on homeostasis. And this slide is here to illustrate that, of course, there were significant differences between the recipient biology in both our study as well as Dr. Montgomery's. And I'm including here a couple of figures from our paper that show the elevations in PTT as well as liver function tests, which you can see depicted here in blue. The x-axis that I'm sure you can't read depicts the time course after transplant, and in short, we know from many different measurements that Mr. Parsons had fairly significant physiologic derangement. We attributed these changes very much so to the mechanism of injury and ultimately the brain death that he sustained as brain death physiology. And you can see from these bullet points below that because of our aggressive attempts to allocate Mr. Parsons' organs, we were only able to perform this experiment, or even sought permission from the family to perform this experiment, until after all of the organ lists had been exhausted. So we began our experiment 5 days into brain death, and you can see here the cause of termination 72 hours later that we reported was due to significant coagulopathy, again, which we attributed to brain death physiology, and ultimately due to hemorrhage. Dr. Montgomery's patients, in contrast, I should say that his decedents appeared to have less physiologic derangement. They were on many fewer pressors than our decedent was. And again, really highlighted up in this panel D here at the top right, you can see for both recipient 1 as well as recipient 2 in the red, that their creatinines pre-transplant, so this is in the first 24 hours before the transplant event shown in by the dotted line, were well below 2, and suggesting that the overall physiology going into their experiment was quite different than our group had. Unfortunately, the mechanism of injury, the duration of brain death prior to transplant, as well as the cause of termination, was not reported in the manuscript. So we look forward to hearing more details to better understand why, for example, some of our results may differ, and how best really to employ the Parsons model and interpret the Parsons model as we move forward, and I'm sure at both our centers with additional decedent studies. A major question that, again, still persists in the field despite all of this work that has been performed and we're showcasing for you today is the question, what are the ideal genetic edits? And in our transplant, in this porcine kidney, we use what we refer to as a 10 gene edited kidney. I'm not going to go through all of these gene edits here in detail in the interest of time, but in short, we included 4 gene knockouts, the 3 carbohydrate antigens that Dr. Locke had emphasized earlier in the talk, which is really to help promote compatibility, as we've discussed. The growth hormone receptor knockout is, we think, quite important to prevent long-term complications in a living recipient, because we do not want growth of the porcine kidney to be out of control and to threaten outcomes after transplantation. We also included 6 human transgenes that essentially modify various aspects of the immune system, as well as, importantly, the complement system, which has been known to promote acute humoral rejection of xenografts, porcine xenografts specifically, in models of non-human primate transplantation. So I think that there is consensus in the field that complement regulation of one kind or another is likely to improve outcomes in human recipients of porcine xenografts. And the genetic modifications that we used in this particular pig, we think are important to control those outcomes later on. Again, that remains to be tested until we actually perform durable long-term survival in human living recipients. Dr. Montgomery's study has already alluded to, you did use a gene-edited kidney, but it was the alpha-gal knockout only, so it was a single gene knockout. And they did not report in their study whether or how they tested to make sure that that particular pig was a alpha-gal knockout. In our study, we reported, with the exception of the knockout for the human growth hormone receptor, the transgene expression, as well as in the donor animal that we used, specifically the presence of the other 3 gene knockouts. Another difference between our studies was that we thought this was critically important to do, was to confirm that there is, of course, our pigs are coming from a pathogen-free facility. Again, I do not expect you to read this table, but it was included in our report. It really showcases for you our testing strategy to survey for a variety of pathogens that could potentially threaten both the pig health and, very importantly, a human recipient health. We have negative testing for PERV-C. There is positive testing in our pig, as pretty much there is in almost all pigs. The positive test for PERV-A and B, and you'll see that result again in a moment. We did not detect, importantly, in light of recent events and recent reports out of Maryland, any porcine cytomegalovirus, and you can see here a negative result by PCR. Now, again, importantly, the Maryland experience was recently reported. Dr. Adams and our moderators have again emphasized how dynamic this field is, with great results and new results emerging all the time. But we knew that our pigs were negative for porcine cytomegalovirus, but did not actually, for the purposes of our publication some months ago, test for this pathogen, particularly in the decedents afterwards. So that is missing from our studies, and we are performing follow-up studies on banked serum samples to be able to confirm that despite the pig testing negative, that, as we would expect, that our human recipient was negative as well. Very importantly, and I think this is my last data slide to share with you before we open it up for discussion, and are very much looking forward to your questions. Both Dr. Montgomery and our group was very interested in studying the potential for viral transmission, specifically of the PERVs, as well as to assay for chimerism. And when I say chimerism here, I mean to be able to detect pig tissue, or specifically pig cells, that may be present in the human recipient in the blood. And just to walk you through this data slide for a moment, and I will spend a little bit of time on this. You can see here that what this is, in our study, what we used here, and actually this was performed by our colleagues, specifically David Ayers and his group at Revivicor, which again was the sponsor and supplier, the creator of both pigs that were used in both Dr. Montgomery's studies as well as ours. These pigs were tested for PERV-A, B, and C, and the results I'm showing here is the gel that is run after the RNA from both human blood and the decedent, as you can see here. I'm sorry, I'm trying to get my mouse to work a little bit better. There we go. Importantly, we have some controls, which this is a positive pig that tests positive for both the porcine, this is a ribosomal protein here that we're using as a measurement of chimerism. The ribosomal protein, RPL4, has allelic differences. It's different sequence, different protein than in the human. So we can detect porcine RPL4 specifically in pigs, but not humans. You can see our donor pig being positive for this. The donor pig over here in this column is positive for the porcine RPL4, but is absent PERV-C as expected. Does test positive, as I already mentioned, for PERV A and B. And again, this is an assay that detects A, B, and C, but again, this is and then a PERV C specific assay here. Importantly, in the decedent blood, including at termination at 72 hours, we see no evidence of chimerism, no evidence of PERV A, B, or C transmission. Dr. Montgomery's group in 2 different recipients really showcase the same results. And this is shown here. You see an absence of bands both for PERV C, PERV ABC, as well as Porcine RPL4. And this is with our reverse transcriptase PCR, as well as no RT-PCR. Importantly, Dr. Montgomery's group was able, with the help of Revivicor, to perform the sensitivity test of the reverse transcriptase PCR assays. In our discussion section at the time of publication, we acknowledged very much that our assays are research and that they are currently under development for detection of both chimerism as well as PERV potential transmission. This group was able to showcase here with the sensitivity test that they're able to, for a fixed amount of RNA from their positive pig, to actually show its dilution over time. And I won't go through the exact precise results of this, but you can see here that roughly around 5 nanograms of RNA, they lose detection in these assays of PERV A, B, and C, a little bit higher for PERV C, and a little bit lower perhaps for their Porcine RPL4 assay. But I very much want to caution the audience here not to overinterpret these results, because while this is evidence that one, of course, can assay or determine for any given assay what the limits of sensitivity are, there are still a lot of questions that remain here. And I would argue that both in our paper and in Dr. Montgomery's paper, this does not represent what I'll call state-of-the-art qPCR testing that will be expected of us as we move into clinical trials of human living recipients. And so while these results are encouraging, and I certainly see that they are consistent, it's very important not to overconclude here, both because the experiments are limited in their duration and obviously cannot predict the rate of transmission of PERVs into a living human, and also because the modalities that have been used to test for these things are quite limited in what I'll call very much first generation tests. So I won't go through all of the text that is shown on this slide, but we've talked a lot today about what knowledge gaps have been filled by these 2 studies. Hyperacute rejection is avoided, and we think that together, despite the different gene edits in the pigs, that the alpha-gal knockout really represents a critical gene deletion that's going to enable the successful xenotransplantation of humans. Multiple knowledge gaps persist. We covered many of these over the course of the data slides. And then ultimately, at the end of the day, we want to know how these studies inform next steps. And I think while there are some significant differences, both in the design and in the findings of Dr. Montgomery's trial as well as ours, that overall, between these and the non-human primate experiments that were critical that came before us, we can all conclude that studies in living humans are essential for many reasons, but primarily because we are looking for additional data that persists out past 72 hours. And while under ideal circumstances, it's very much possible that a decedent model could persist for a longer period of time than either 54 or 72 hours, we probably can all agree that many of the questions we want to answer, particularly regarding PERV infection, for example, are not going to be questions that we can answer in even a longer term decedent model. We need to move into living humans, and we need to do this as safely as possible. And again, I think the field is excited because we think that in total, the studies today performed in both the decedents as well as the trial that we're awaiting publication from the Maryland group and the heart recipient are very much clear indications that many of our initial safety concerns and questions have really been addressed. So it's an exciting time to be in xenotransplantation. So of course, we want to acknowledge our group here at UAB that did the work. Very, very importantly, this the slide here represents just a fraction of the people who participated in our trial. Our study group was well over 100 people, and we're just showing here the faces of our surgical team that participated both in the procurement as well as in the recipient transplant. And with that, Dr. Locke and I are really eager to answer questions. I apologize if I've gone over a little bit of time. It doesn't look like it.
No, that's great, Paige, Jayme, right on time. So we have probably 15, 20 minutes or so for some discussion, question and answers. And if you have questions, I think Brian has kind of mentioned this in the chat, but please enter those in the Q&A button down at the bottom of your Zoom. So Burcin, you want to start out?
Sure. Again, thank you very much. I first like to say that I was warned that I don't have any conflict of interest because I didn't send those since I didn't have. So thank you, Brian, for reminding. The first question from the chat, actually, regarding this Parsons model you mentioned at UAB. So we are very thankful to the family, of course. So how does the FDA view the Parsons model? Will evidence from the brain dead individuals be included in IND submissions, since you guys have any the experience with IND?
Thank you for that question. I certainly don't want to speak on behalf of the FDA, but what I would say is that absolutely, I think data from preclinical human model, the Parsons model, is going to be very critically important in any submission to the FDA, along with all of the non-human primate work. I think the full body of work is going to be necessary to really sort of be able to demonstrate to the FDA what the field has done, what questions have been answered, and what remain. And the questions that remain, I think again, don't want to speak for the FDA, but I think any regulatory body wants to ensure that there's not some other way to answer those questions prior to moving to living person. So I think it's going to be really important to share all the work that has been done in this regard.
Great. I think next in the list there, Dr. Reese from Penn had a question I think Paige you partially answered. He was asking, could you please review the genetic modifications made between the two studies, which I think you did a great job in your chart. Then his second part of the question was, and if possible, make connections between those modifications and the clinical outcomes observed. And I'd add on to that, and you started this discussion too, where it's, I think, an overarching question of what, and Peter is just chatting up that the Maryland heart, I think, was the same pig that you guys used, correct, Paige and Jayme?
Yes, correct. To our knowledge, it was a 10 gene modified pig, also supplied by Revivicor.
Yeah. And then I guess the question is really just a comment on what do we think is necessary, because that's the, $1 million question, which genes there, you can make a theoretical kind of argument and there's data supporting each of those pathways, which is, clear. And, but then surprisingly, again, very short-term outcomes with Dr. Montgomery's study versus yours in the Decedent model, how do you kind of reconcile those two things with, very minimal modifications? And I don't think and I haven't really delved into his whole paper about did they do any complement, therapy, anti-complement therapy there. I didn't see anything in the methods, but yeah, maybe just comment on that with regards to Peter's question, genetic modifications, outcomes. How are we going to determine which modifications we need?
And we'll tag team that. I mean, I think, I think that is obviously a topic of great debate. If you look at some of the previous work that's been done, without question, the compatibility in humans increased as you progressed from knocking out one xenoantigen, knocking out 3 xenoantigens, you see the compatibility improve with that. So if you're looking at those data, I think you could make the argument that in fact you should have all 3 of those knocked out. In terms of comparing it to the function, I think both studies are limited in that regard, depending on how you describe function. I think what both studies very clearly were able to demonstrate is that there was no hyperacute rejection. In our particular study, Mr. Parsons had been brain dead for 5 days. He was on 3 pressors at the time and was already in DIC when we actually did the transplant. So I think that's a pretty hostile environment, and so it's unclear, to really be able to claim clearance or to look at those kinds of things in that model, I think is quite challenging. The important piece is that the native kidneys were removed. So I think if we can find a preclinical human decedent that's more stable with the native kidneys removed, you may potentially be able to actually assess function, both in terms of volume of urine as well as clearance, because urine does not equal clearance. In the Montgomery study, the native kidneys were left in place, and the transplanted kidney was actually implanted in the leg. But importantly, the native kidneys were left in place, and because they didn't isolate that, it's really, I don't know that you can claim that the xenograft actually was responsible for the decline in creatinine. That change in creatinine could simply have been as simple as hydration. And they, to their credit, acknowledge that very blatantly in their results. And they say you can't actually derive any conclusions about kidney function from their model. So I don't know that you can really say at this point which one did a better job in that regard. And in terms of the biopsy findings of us having TMA and them not, again, without more data from their model, it seemed that their donor was much more stable and not in DIC at the time of transplant. Ours was, and we saw this fibrin thrombi, you know, in the xenograft. So unclear. It could have just simply been from the brain death milieu. And Andrew, I don't recall that they used a complement inhibitor in their paper. But so all that to say, I mean, I think you've got two kidneys, the U kidney and the U thymo kidney, and neither had hyperacute rejection. I think that is what you can conclude. And I think the other advance is that we were able to develop a flow cross match that works a priori. So you should be able to predict ahead of time that you're not going to have hyperacute rejection. I think that is the most significant piece. I think sorting out creatinine clearance has got a ways to go. And as best I can tell, I don't think they measured excretion of creatinine in the urine to be able to sort of attribute any of that to the xenograft itself.
Just a couple of comments. Dr. Locke obviously just described the multiple variables, you know, variations, differences between our two studies. And so that really confounds any conclusions. We cannot attribute any of the outcomes that we observed to specific gene modifications or lack thereof. I do think that this is one way to look at this, is that we really do, again, need trials in living humans with longer term results to really answer these critical questions and gather the data. And I think that one other way of looking at this is that, again, I will not speak either for the FDA, but there's no reason at this point in time, I don't think, unless there is a serious safety concern with one particular design or another, that we shouldn't encourage multiple different submissions to the FDA of multiple genetic edited pigs, of different types of immunosuppression regimens, because we aren't going to know yet what outcomes are associated with any one of those variables. That's going to be information that is yet to come.
I also have I'd like to tag along to this function of the xenotransplanted kidney, because I know that you guys cannot speak for Dr. Montgomery's behalf, but why didn't you choose to do a renal scan? So you could just estimate the GFR, and as well as also the NYU study, because the native kidneys were left, just doing a simple renal scan, at least the estimation. Why didn't you choose to do that?
It's a great question. I can't tell you how badly I wanted to do that. And I'm sure Dr. Montgomery would probably give you the same response. You know, the biggest issue is that right now everything has to be very contained. So because, you know, you have to make sure that you're not putting things back in circulation. So similar to NYU, all of the equipment, everything we used was unique to this study. It was not in circulation with any other part of the hospital or clinical care. So moving the decedent to an area that has living persons with a genetically modified pig kidney that has been transplanted in them is really sort of, I think, was not something that we were interested or wanting to do. And I think our regulators here at UAB and the regulators at NYU certainly agreed with that as well. So I think until we have an actual IND with a surveillance program in place that the FDA is happy with, I think having these individuals move throughout the hospital is going to be a bit challenging.
Just a follow-up question that came through with regards to kind of the fibrin thrombi or TMA. Did you have the opportunity to look at biopsies from Mr. Parsons' native kidneys or liver or some other organ that might give you a clue as to maybe the driving force behind that?
Yeah, great question. Short answer is yes. We did have access to the native kidney biopsies from Mr. Parsons, and have done additional work to study whether there's TMA in other organs, as you mentioned, Andrew. And in short, we didn't observe any of that. So we were hopeful that we would, because that would obviously have given evidence of potentially the contribution of brain death and brain death physiology to the TMA we observed, but we did not clearly see it.
Another theme that came through in a couple of questions. So regarding the transmission, and again, not either one of these studies, but the transmission of porcine CMV in the report, at least that came out in the the comments by Dr. Griffith and Dr. Moedine. So part of it was the question is do we feel like this significantly sets the field of xenotransplant back, or can porcine CMV be knocked out? So maybe what your strategy is, or what the field strategy is for preventing, in particular, porcine CMV, which at least experimentally has been shown with a higher incidence of rejection in preclinical models. And I think Dr. Rothstein had a similar question.
I think it's a great question. So primarily, the animals have been surveilled using PCR. So you actually have to have circulating virus for that to actually pick up anything. But we know that CMV is a latent virus. And so really, I think an antibody assay is going to be necessary. I think that's something that is a gap that the field's going to have both to surveil the pig, but also to surveil. So I think that's going to be super important. And that may have been, what happened in the case of the heart patient. I don't know. It could have been, I don't know the CMV status of their pig, but if the PCR was negative, it is possible that the pig had it and had antibody to it. But if that wasn't detected, then you wouldn't realize that. And then you transplant that in the setting of immunosuppression, and you can certainly have the reemergence of a latent virus. So I think that is something that's going to have to be addressed. And I can't imagine that federal regulators won't require it.
And another comment there is that, Andrew, to something you just said about whether porcine CMV needs to be engineered out, as that's an approach, obviously, that some companies have taken to call it control or limit the potential for PERV transmission. I think the good news is that we don't need to engineer out porcine CMV, because it is possible to exclude it from the colony. But just to echo Dr. Locke's comments, I think this is the perfect example of, frankly, and I use a funny word here, I feel like we've been a little bit bamboozled, right? Everyone in microbiology and immunology and medicine, we really love our PCR tests. We're very, very good with them. But I think this is a classic example of how perhaps we did not think very closely about the lesson we learned about latent CMV transmission, right, in human allotransplantation. I think all of our centers probably collectively do the same thing. And in our deceased human donors, which is we do not test for CMV viremia with PCR testing, we do serologic testing to see if there has been exposure to that virus in the past. And so, again, this is an example of the must-haves, in our opinion, moving forward, which is serologic testing to accompany the PCR testing specifically for this virus. Big question is, what else do we need to survey differently? And I think this is a really great opportunity for the community at large to weigh in on this and think more about and demand more from the tests that we are providing before we move into humans, living humans.
Yeah, I think that's a good point. And for Dr. Rothstein, many of these pigs are derived via cesarean section and kept in, quarantine herds that exclude CMV. Yes. But just like for humans, CMV is fairly ubiquitous in the population. And so having the ability to detect that ahead of time, and whether or not that contributed to any of the outcomes in the heart transplant, we don't know. So, Burcin?
Yeah, as you mentioned, UAB is now famous with this transplant, and then I know that you guys published also some non-human primate studies since 2016. We have also seen the importance of the growth hormone knockout pig, especially after the German group's paper in Nature in the heart transplant in baboons. And Maryland group used that genetically engineered pig, and then you said that you used it, even though you didn't confirm you used it. Do you have any experience with the preclinical kidney xenotransplant in non-human primates? Have you used those pigs?
Yes, we have, and definitely want to give credit to Dr. Cooper and his team, Dr. Yamamoto, who I think had a question. When they were here at UAB, they really drove our non-human primate work, and we do have an experience using the 10-gene edited pig in the context of a non-human primate model. And certainly don't want to put words into Dr. Cooper's mouth, but I know he had experience using the genetic edited pigs both before and after the growth hormone knockout. And the growth hormone knockout turned out to be quite important to control the size of the kidney. And we have some data that I didn't show, hasn't been published yet, but that just sort of shows that knocking out the growth hormone really does prevent that exponential growth. And at least from what I can tell, I think the pigs used for the NYU experiment and the one used in ours were roughly around the same size. And you can see how much bigger the kidney is, at least it looks like in the picture. I don't have the measurements, but just eyeballing it looks bigger. So that growth hormone knockout, I think, is going to be really critical in the pig.
Yeah, the reason that I was asking, because this is only 2-3 days study, so we cannot actually see the difference of growth hormone. But if you have that in the long term, at least 3-4 months or maybe 6 months, I don't remember. So it might be, so thank you for responding. And definitely, I wasn't ignoring Dr. Yamamoto's question. So he asked that question regarding C4D staining. And if you had, because we all know that pig studies are very difficult, if you had any positive control for C4D staining.
He's asking.
Yeah, our laboratory definitely runs positive controls with whenever C4D staining is performed. But to your point, the question is, did we have a pig kidney, right, as opposed to a human kidney that tests positive for C4D? And the answer is no. So our positive and negative controls were, I call it standard issue from our laboratory in terms of making sure that the IHC results were correct or interpreted as non-specific. So I think it does beg the question of whether how one interprets C4D staining, and if there's perhaps a difference in the type of methodologies that I use to detect it. But as with so many of our histologic as well as other types of pathologic tests that we use, everything comes down to the controls and the interpretation. And rarely ever are things so black and white. Everything is a shade of gray, right? So anything we can do to, call it, improve these tests and to make them more quantitative is going to certainly be valuable, at least I think so, in the future.
So we have time maybe for one last question. Take Dr. Voss's question, and hopefully it's not a lengthy answer, but it could be a challenging one. So she asked in her second question there about the difference in immunosuppression between, say, what was done in the heart transplant model using a novel co-stimulatory blockade reagent, an antibody against CD40, versus the strategy that both you and I think Dr. Montgomery had with more standard immunosuppression, and what the thought is there, what you think in a clinical experience where longer term outcomes may need to be or will need to be monitored, kind of what the approach will be.
So I can take that. I think that the co-stimulatory blockade is really born out of the non-human primate work, and the data in non-human primates clearly shows better outcomes with the use of that, particularly in the context of heart. But when we sort of looked at those data, again, all of those transplants, for all intents and purposes, are being done across incompatibilities. They're being done across a positive cross match. And in general, immunomodulation prior to the transplant really doesn't occur. So you're not plasmapheresing these animals. I think there's historically people in the past have done some immunoabsorption, but it's not something that's been done in the recent present. So if you really want to kind of get past an early issue, I think you have to give a ton of really significant and powerful immunosuppression up front. And we just felt that in the context of using a compatible transplant, that we should be able, ideally, to use standard immunosuppression. And importantly, wanted to be able to study an immunosuppression regimen that's already approved, so that we're not trying to sort of introduce another agent that would be under the context of an IND. So I hope that wasn't too long.
No.
And just one last quick comment about that is, again, based on what I said before, I think there is room for everything. We don't know the gene edits. We don't know the ideal immunosuppression regimen, which may absolutely vary if we do a compatible or an incompatible xenotransplant. So I think all of this work just showcases how much more there is to do. And we are simply not going to know the answers to these questions for a long time. I don't see any a priori reason why we should not restrict moving into humans to any particular gene-edited pig or any particular immunosuppression regimen.
Yeah, well, that's it. On behalf of Burcin, I'd really like to thank both Jayme and Paige for the great work they're doing. I think the stimulating discussion we had between both their work and recently Dr. Montgomery's work, and really laying the foundation for work moving forward. Burcin, you have any parting comments? Otherwise, we can turn it over.
Oh, you guys all wowed us very positively because you jumped in the xeno field from nowhere and very positively, believe me. And a lot of colleagues asked us from Asia, Europe, okay, who are these people? And then they are publishing, and so we look forward definitely to read more from you guys and contribute more to the xeno field, because as a part of IXA also, that we are very excited to read your research.
Well, thank you for having us. We hope we can contribute. We appreciate it, y'all. Thank you.
Excellent. AST would like to thank Drs. Porrett, Locke, Adams, and Ekser, and all of our attendees for today's session. Please remember to complete the evaluation survey and visit myast.org/journal club to view our video archives and register for upcoming AST AJT Journal Clubs. To learn more about the AST communities of practice, please visit myast.org/COPS or connect directly to the KPCOP hub. Thank you all again for today's excellent session.